Health is an eternal topic for mankind. We are born on Earth, and our health is intimately linked to the environment and ecology. The environment is the basis for human survival and development, and every subtle change in the environment will eventually have an impact on biodiversity, ecosystem, and our health. Exploring how the environment and ecology affect our health in the past, present, and future is the mission of Eco-Environment&Health. Eco-Environment&Health is a journal initiated by prestigious scientists with a strong editorial board, including multidisciplinary scientists from all over the world. With the support from EEH, you will have a better publication experience. EEH has its own offices of publishing, language, design, and broadcasting. The experts will accompany you like an harmonious band and spare no effort to sublimate your valuable scientific research into a magnificent and impactful masterpiece. We have a dedicated center for decision-making that deeply integrates informatics, ecology, and environmental science. It explores global research hotspots, predicts discipline trends, monitors the influence and dissemination of publications, and supports the journal's smart decision-making. Upon receiving your submission, our assistant editors will contact you immediately and keep you informed of the manuscript status throughout the entire process. The 14-day rapid peer review will make the publication of your research one step ahead. Your distinguished work is worthy of being known worldwide. We will promote your research globally in a three-dimensional manner through various social media platforms. In addition, the open access fee will be waived for the first 3 years, making your research easier to be read and cited by your peers. We continue to explore and innovate the service strategy of the journal. EEH leads ecology and environment to a healthy future.
Hello,
okay,
Dr. Shen.
Yes, I can hear you. Thank you, Doctor.
Could you please begin your presentation?
Yes. Thank you, Dr. Mo, for the very comprehensive introduction of me. And thank you, EEH, for the invitation to share my work. And thanks everybody to taking the time to come to my seminar tonight at 9 p.m. In this talk, I will talk about my research on themed harnessing high-dimensional exposure and omics data in environmental health sciences. And as introduced, my name is Shen Yike. Okay. So I want to start my talk by my perception of environmental health data science. It consists of the understanding of the high-dimensional exposure, omics, and various health outcome data. We as humans are exposed to a myriad of chemical pollutants, for example, the pharmaceuticals and the personal care products, metals, air pollution, pesticides, etc. Those chemical exposures that can collectively alter our epigenome, metagenome, metatranscriptome, or sometimes we call all of them ends with omics as the omics, and in turn change our health status. My research focusing on deciphering the relationships between the exposure, omics, and health outcomes. So today, I will first discuss my research linking the environmental exposures and the microbiome in both plant, soil, and human settings. And I will then talk about our recent advances in developing the novel deep learning models to predict chemical ecotoxicity from high-dimensional physiochemical features. And I will also talk about our advanced machine learning models to predict fate and transport of pesticides using the chemical molecular fingerprints. So stay to the end for our department's new MS program in environmental health data science information. Okay, now let's move to the first part, linking the environmental exposures and the microbiome. So I will start the exposure of antibiotics in relation to the plant soil microbiome. The increased anthropogenic antibiotic production and use in the animal feeding, aquaculture, and human treatment, as well as the waste, have led to an increase in the trace level of antibiotics residues in the reclaimed water, or sometimes we can call it reused wastewater. Due to the global water scarcity, more farms are using the reclaimed water for crop irrigation. So the irrigation water that contains the trace level of antibiotics could amplify the antibiotic resistant genes, as shown as antibiotic resistance, as shown by the antibiotic resistant genes, or sometimes we call them ARGs, and bacterial communities. So in my doctoral studies, I have designed and conducted the laboratory experiments studying the antibiotic resistant and the bacterial microbiome in the lettuce soil system in response to antibiotic exposure. Now the sample name starts with C on the left, are the control samples without the pharmaceuticals. Next to it are the samples irrigated with water containing a mixture of 8 antibiotics and 3 non-prescription drugs at the environmentally relevant concentration. The antibiotic resistant genes profiles are showing on the left, and the bacterial community are showing on the right. So the antibiotic resistant gene profiles and bacterial communities have shown the changes in the relative abundance of the ARGs and bacterial community with the pharmaceutical exposure. For example, some of the mobile genetic elements, those elements are capable of transferring from one bacteria to another, such as the integrase 1 highlighted here, showed an increased relative abundance with antibiotic exposure. So bacterial family of Macellophyllaceae, which showed here in brown, showed the increase in relative abundance with the pharmaceutical exposure. So this family belongs to phylum Proteobacteria, which are the most susceptible phylum with the bacterial species that are capable of horizontal gene transfer. So from the previous study, we found that different compartments showing different alterations of ARGs and bacterial community. Researchers have shown that plant roots to have a strong control over bacterial community. So I wonder if the plant roots associated soil bacteria, aka the rhizosphere bacteria communities, shows the difference with the other part of the system facing the antibiotics exposure. So I therefore conducted a growth chamber experiment, irrigated the lettuce with a mixture of 9 antibiotics from major antibiotics classes. And then in the results showed in the network figure, the red nodes are the antibiotics, green nodes are the bacterial communities, green nodes are ARGs, sorry, purple nodes are bacteria. So indeed, the rhizosphere and roots, as shown on the left, had a high bacterial community interactions and ARG exchanges. The most mobile genetic elements were found to be hubs in the network that plays an important role in the rhizosphere soil. So given what we found in the lettuce soil system with the trace level of environmental exposures and the importance of the host, I wonder what changes will the chemicals induce in the human gut microbiome. So that's when I started my microbiome epidemiology studies in my postdoctoral training. So the gut microbiome is important in modulating human health. So the gut microbiome can influence human physiology by the host digestion and the nutrition absorption. Also, as well as the complex and the bidirectional microbiome host relationships in the immune systems. Also, the microbiome was known to change associated with diseases. For example, the inflammatory bowel diseases, the IBD. And another hot topic now is to study the gut-brain axis, basically the gut microbiome changes and the neuropsychological outcome of people. Okay, so the gut microbiome is important in modulating health in childhood. As we all know, metal exposures affect multiple health outcomes, but interestingly, their ability to modify the bacterial community in children is poorly understood. In addition, there are also very limited human studies to assess associations of metals or pharmaceutical exposures to the pediatric microbiome. So in my first microbiome epidemiology study, we aim to assess the associations of childhood and perinatal blood metals with childhood gut microbiome diversity, structure, species, and potential pathway alterations. Our study was performed in the gestation and environment cohort in Sherbrooke, Quebec, Canada. So the GEST cohort is relatively homogeneous in demographics, consists of primarily upper to middle class French Canadians with levels of exposures comparable to US population geometric mean. We included two sets of participants. The first set is the prenatal exposure to child microbiome, 70 participants. And the second set is child exposure child microbiome with 68 participants. The blood metals were measured in mothers at the delivery and the children at the 6 to 7 year old follow-up. So DNA from the stool samples in the 6 to 7 year old follow-up was extracted and sent for the 16S amplicon sequencing and the shotgun metagenomic sequencing. So the raw data were batch processed in a supercomputing cluster using CHIME2 and Human3 pipelines, and later analyzed for the diversity, phylum, family, species, gene family inferred species, and the pathway associations. All of our models have adjusted for the different covariates set. The child exposure, child microbiome is adjusted for sex, family income, gestational age, and ever breastfeeding. The prenatal set is adjusted for sex, family income, delivery mode, maternal BMI, and pregnancy complications, and the pre-existing conditions. This paper is published in Environmental Health Perspective in 2022, January issue. So moving to the results, we found no significant associations of both alpha and beta diversity with either prenatal or childhood blood metals. The previous animal studies on metal exposure and gut microbiome diversity have yielded conflict findings. The human studies of metal exposure and the gut microbiome among the school-age children have been extremely limited. The only study we found on metal exposure to the 3 to 7 year olds was heavily focused on the autistic children without analyzing the metals in a continuous manner and childhood gut microbiome diversity in the continuous fashion. So our study in adult found that the gut microbial evenness increased during a long-term lead exposure near a mining site and a smelting site. This study was actually conducted in China. So our study suggests that influences from prenatal and early life metal exposures may not have a long-term influences on the diversity of childhood gut microbiome. So now moving on the tax and pathway associations. We found both the long-term and the short-term associations between the metal exposure and the childhood gut microbiome, with a stronger association observed with more recent exposures. So for all significant associations, I select a few to talk here. More information on them can be found in the paper. So the higher childhood blood manganese was significantly associated with lower in vitro bacteria, which was found to associated with the secondary plant compound metabolization, which also mediates some of the health effect associated with the fruit consumption. We also found that higher childhood blood selenium was significantly associated with a higher relative abundance of the proteobacteria. So as I previously mentioned, I also found the proteobacteria increased in the relative abundance with the environmental stress, such as the antibiotic exposure. Some of the proteobacteria members have been in line with multiple negative health-related outcomes, such as the dysbiosis and metabolic diseases. So lastly, the higher childhood blood lead was also significantly associated with a lower relative abundance in the acetylene degradation pathway. However, we do not have an immediate explanation of why this pathway result came out. Okay, so moving to the gene family inferred species association. So the gene families are a group of evolutionarily related protein coding sequences that often perform in similar functions. The higher relative abundance of the potential pathogens, beneficial species, and both potentially pathogenic and beneficial species was found to with the higher childhood or prenatal blood, cadmium, mercury, and lead. So showing in the red here, most of the bifidobacteria members are the beneficial bacteria that establish the metabolic and physiological interactions with the host. So in particular, the bifidobacterium longem has a high adaptation with the human GI tract. It also have a potential immunological activities and may be used as the probiotics to reduce adverse health outcome. So in fact, lots of probiotics are from the bifidobacteria family. Okay, so this beneficial species may have adapted to the population blood metal level, and thus have a higher relative abundance in relation to higher blood metals. So moving on to the Bacteroides species. So the Bacteroides are the obligative anaerobes that primarily in the human lower intestinal tract that regulate the molecular interactions. The Bacteroides are species are friendly commensals and they're ubiquitous in human gut. For example, the Bacteroides uniformis has been shown to mitigate metabolic disorder and immunological dysfunctions. However, the Bacteroides can be harmful outside the human gut environment and was found in most anaerobic infections. So lastly, the Flavobacteria polotii, we're showing in black here, a potential pathogen with a significant positive association with the childhood blood cadmium, was also assisted with worse social responsiveness scores and was found to suppress the immune response in mice. So in conclusion, our study found that the associations of childhood and the perinatal blood metals with relative abundance of specific gut microbiome taxa and the gene family in first species that based on the previous findings may be beneficial to the host, while others may have negative consequences. We recommend additional epidemiological and mechanistic studies to confirm and generalize our results. Okay, so different blood metals in healthy individuals can alter gut microbiome. So I wonder if the perinatal exposure of commonly used pain reliever, the acetaminophen or Tylenol, and caffeine can have a long-term effect on the childhood gut microbiome and their subsequent interactions with the neurodevelopment. So pregnant individuals are commonly exposed to the acetaminophen to relieve the pain and of course the caffeine, but it is unknown that how these exposures interact with the developing gut microbiome. So in this second microbiome epidemiological study, this is a collaborative work with Dr. Hannah Lau from Dartmouth College. We aim to determine whether acetaminophen and caffeine relate to the childhood gut microbiome and whether features of the gut microbiome can alter relationships between the acetaminophen, caffeine, and neurodevelopment. So in the same Canadian gestation cohort, we measured the meconium concentration of caffeine and acetaminophen. I want to mention for a second the meconium here. So meconium means the first stool of an infant. It begins to accumulate in the second trimester of the pregnancy and can capture the cumulative exposure over the last 6 month period in mothers, making it a perfect matrix to capture the prenatal long-term exposure. So the meconium concentration also helped us to reduce the conventionally called recall bias, where mothers were asked if they have taken acetaminophen or caffeine. we directly measured the concentration. We also collected the neuropsychological questionnaires, including the WISC, which is an IQ test, and the Q-TEC scores. So there are approximately half of the participants with acetaminophen not detected. We therefore dichotomized acetaminophen to detected vs non-detected. The microbiome data was sequenced using the shotgun metagenomic sequencing and followed the same raw data processing workflow as the previous study. So our results showed that the acetaminophen, but not caffeine, was associated with the differences in childhood gut microbiome. So specifically, the prenatal acetaminophen was associated with a lower alpha diversity. It's accompanied by the decrease in firmicutes and the increase in actinobacteria, and those genes in the pathway of succinate fermentation to butynate. We found no association in the species level. So the higher taxonomic diversity is generally thought to indicate a more mature system, as it contains a greater functional diversity to complement the host response to exposure. So infants have low taxonomic diversity and later increase with the introduction of the solid food. So similarly, the vaginally delivered infant was found to have relatively low levels of firmicute and high levels of actinobacteria compared to later samples. So it may be possible that the prenatal exposure to acetaminophen delays the maturation of the gut microbiome to a more adult-like composition. So we further tested if there's any interactions between the gut microbiome and the neuropsychological outcome. So we tested whether the most abundant, which is the top 10% of the phyla species or pathway, modified the associations between exposures to acetaminophen and caffeine and neurodevelopmental scores. So we found that the relative abundance of proteobacteria modified the neurodevelopmental effect of both acetaminophen and caffeine. So the left panels are acetaminophen, right panels are the caffeine. So the participants with detectable concentrations of acetaminophen had a slightly higher scores on the WISC subsets, which is the IQ scores. But this benefit was weakened among those with the higher level of proteobacteria. So in contrast, higher concentration of caffeine was related to a higher scores of several WISC subsets. But this effect was exacerbated among children with higher levels of proteobacteria. So proteobacteria, as previously discussed, include many notorious pathogens. And in addition to the 3 previous studies found changes in the proteobacteria with antibiotics and metal exposure. Studies also found the elevated relative abundance of the proteobacteria is associated with the worst cognitive performance or impairment in clinical population-based and animal studies. So our findings suggest that in addition to the direct negative relationships between the proteobacterial relative abundance and the cognitive scores observed in the previous studies, it may modify the association of caffeine and acetaminophen with the cognition in an adverse manner. So in conclusion, in this study, we identified an association between the prenatal exposure of acetaminophen and gut microbiome composition in the mid-childhood. We recommend larger studies to confirm this result and examine the mechanisms by which the in utero exposure affect the long-term gut microbiome development. Additionally, we found the evidence in this study that certain bacteria may modify the relationships between the exogenous exposures and the health outcome, a potential explanation for the previous discrepancies in the previous studies on these exposures. So overall, this study positions the microbiome as a critical modifiable feature in the future environmental health studies. So to recap what I just talked about, the environmental exposures altered soil, plant, and human gut microbiome. All 4 papers that I have discussed is published and can be found on my website at equation.github.io. Okay. So in addition to linking the environmental exposures and microbiome with statistical methods, another aspect of my research in environmental health data science focuses on developing machine learning and deep learning models in predicting chemical toxicity and disease. So in the Environmental Health Center, Health Sciences Core Centers, or EEHSCC meeting in this July in New York, the NIEHS director, Dr. Rick Warchard, talked about the NIEHS initiative on the environmental health data ecosystem. So with this initiative, we are going to incorporate more computation models in analyzing the environmental health data. So in the next part of my presentation, I will talk about how we use machine learning and deep learning to achieve these goals. So specifically, I will discuss two studies on the computational toxicology. One study on developing novel deep learning models to predict chemical toxicity from high-dimensional physiochemical features. Another study is about our advanced machine learning model to predict fate and transport of pesticides using the chemical molecular fingerprints. I will touch our work on the precision environmental health and another study of data-driven discovery. However, we won't have the time to go through the last part. So this part of the research is a major collaboration with my colleague, Dr. Feng Gao. Okay, now let's move to this part, predicting chemical ecotoxicity. So the first, this first study about computational toxicology, we predicted the chemical ecotoxicity. So chemical hazardous concentration or chemical ecotoxicity HC50 is derived from the geometric mean of the effective concentration 50 and the lethal concentration 50. So evaluating chemical ecotoxicity is a valuable approach to identify potential hazardous effect of chemicals to the ecosystem. So the traditional gold standard way of determining HC50 uses in vivo animal models. But those approaches present the challenges in the face of rapid increase in the number of chemicals manufactured each year. So to tackle these concerns, the US federal initiative proposed a high throughput in vitro chemical screening to effectively identify hazardous compound, also known as the toxicology in the 21st century, or TOX21. So however, this approach still requires labor-intensive laboratory procedures that cannot keep up the pace with the manufacturing of the new chemicals. And in addition, current in vitro methods cannot capture the complex effects such as the endocrine effect and have limited ability to predict toxicity in the in vivo system through the method known as in vitro to in vivo extrapolation models. So therefore, the chemicals must also go through the in vivo animal testing and to further determine HC50. So all of this highlighting the importance of the accurate in silico computational chemical screening in the first step. So the most of the conventional quantitative structure activity relationship models, or known as the QSA models, are linear and are not very effective in capturing more complex nonlinear relationships. So the machine learning models have been developed to capture these nonlinear relationships in toxicity prediction. However, not all machine learning models can capture the high dimensional features. So the redundant features in the high dimensional data may introduce noises and increase the difficulty of handling many features. We developed a model called auto-encoded deep learning model to solve this issue. So in the study, we aim to develop the auto-encoded model to learn the chemical latent space representations and predict the HC50 from the in vivo, which is animal models in the US ETOX database, containing over 1,800 chemicals, and there are approximately 700 chemical features. So specifically, we aim to utilize this model to learn the low-dimensional latent space representations of those 700 chemical features for the dimensionality reduction and to improve the prediction accuracy of the HC50 by our model. So what is autoencoder model? Autoencoder is an unsupervised representation learning method that can perform the nonlinear dimensionality reduction. So instead of aiming to preserve the distance of the local structures, it learns the latent space embeddings. And those embeddings contains representation of the data that are more meaningful or comprehensive than the predefined distance, which is usually used in the models of PCA. So the autoencoder consists of a pair of encoders and a decoder that can be parameterized through a neural network shown here. So encoder reduces the high dimensional input features to a lower dimensional embeddings, while the decoder reconstructs the input features back to the back from the lower dimensional embeddings. So this encoding decoding process enables the reduction of the dimension by compressing and removing noises while still preserving the essential information for feature reconstruction. So the low dimensional space embeddings are learned from data and retrieved by minimizing the reconstruction loss. Then the learned latent space embeddings can be used for various downstream tasks, for example, the supervised classification or regression tasks and unsupervised clustering tasks. So the t-SNE plots, t-SNE provides a tool to visualize the high dimensional data. So as shown on the left, the t-SNE plot, it embeds in the local structures of the data and attempts to extract a clustered local group of samples. So the embeddings themselves reflected the patterns of the HC50 value, with each point representing one of the chemicals in the data set. So the chemicals with similar embeddings are supposed to should be near each other and thus have similar HC50 values. So in our results, there was a clear pattern of change from the smaller HC50 value, which is showing in the darker color here, to a larger HC50 value in the brighter color. So the ability to reflect the HC50 value based on the chemical learned embeddings suggests that our autoencoder method is effective in learning the low dimensional latent space representations from of the chemical HC50. So in order to compare our autoencoder method with other dimensionality reduction method, we compared them with a traditional PCA, kernel PCA, and UMAP method. Our results performed much better than all of other dimensionality reduction methods because it benefited from learning the latent space representation of the chemicals from our data. So in order to compare our autoencoder prediction results with other machine learning models, we compare them with the random forest model, fully connected neural network, lasso regression models. So although the R-squared value of the random forest model was comparable to that of our autoencoder model, its MAE and RMSE, which is the error term, were worse. So notably, I want to mention here the prediction of our autoencoder methods was solely based on one simple linear layer from the embeddings, in contrast to a complex random forest model. So this result further highlighted the benefit of the chemical ecotoxicity prediction based on learning chemical embeddings rather than on raw chemical properties. So let's move to the last part of the fate and transport of contaminants in the environment. So the planned uptake of antibiotics have raised serious food safety concerns, and the measurements and prediction of the antibiotic uptake on the plants are often based on the root concentration factors, also known as the bioconcentration factors. So in one of my previous experimental results, it showed that there are significant differences between the rhizosphere soil and the bulk soil root concentration and the bioconcentration factors. So this emphasizing the necessity to design a multi-pollutant and a multi-compartment contaminant fate and transport model when we're studying those questions. So I'm going to talk about the feed and transport of pesticides in the environment in terms of their dissipation half-lives as an example. So the pesticide dissipation half-lives in plant is an important factor to assessing the environmental fate of pesticide and establishing the pre-harvest interval that is critical to good agricultural practices. However, those empirically measured pesticide dissipation half-lives are highly variable, and the accurate prediction with the model is challenging. This considering hundreds of pesticides active ingredient and over 100 of plant types, together with various environmental factors that can potentially result in the tens and tens of thousands of crop pesticide combinations. So previous studies focused on predicting the pesticide dissipation half-lives using the geometric mean, which may over or underestimate the pesticide dissipation half-life intervals due to the variability. So we propose an alternative way of predicting an interval where the geometric means could fall into. So we developed the machine learning models to predict the pesticide dissipation half-life intervals using the molecular fingerprint of pesticides. So what is the molecular fingerprint, or we call it ECFP? So the ECFP is a typical circular topological fingerprints that encoding molecular structures into a series of binary bits. And this is previously developed for the structure activity relationship modeling. So each bit represents one substructure, and it describes the presence and absence of a certain chemical structure. So these molecular structures can well describe specific substructures in the molecules and connecting the molecular properties with the molecular substructure. So a diameter of 4 means working 2 steps surrounding an atom. So the ECFP gives a more objective feature selection, thus avoid manually select pesticide features. So we use the ECFP in combination with the temperature, plant type, and plant component classes as the representative input features into our machine learning models to do the prediction and find important pesticide substructures. Our model was trained and validated and tested in a data set of 311 pesticides and so over 1,000 data points. We used 4 models: the gradient boosted regression tree, the supporting vector machine, random forest, and linear logistic regression as a baseline. So first we use using the k-means clustering, we found that the ECFP of diameter 4 that effectively reflected the differences in the molecular structure across various pesticides through 4 classes showing here. So the group 0, showing in yellow, reflects the difference in molecular. It contains the purely parasitoid insecticide. The group 2 mainly consists of the triazole fungicide and all demethylation inhibitors. The group 3 are mainly the organophosphorus pesticides that contains the functional groups such as organophosphate, while the group 1 and 4 are a mix of the different pesticide categories that share the similar structures. So our result did effectively reflect the differences in the molecular structure using the ECFP. So moving on to the prediction results. So the F1 scores are a common matrix used to evaluate the prediction performance for the classification. F1 microscore didn't account for the label imbalance, while the F1 weighted score account for the label imbalance. So our gradient boosted regression tree model, which is the GBRT, have achieved the best performance. So although the GBRT and random forest are both tree-based models, the gradient boosted regression tree aims to minimize the loss function by building one tree at a time, whereas the random forest randomly sample the data by training each chain independently. So the better performance of the GBRT and random forest may result from its ability of step-by-step learning that strengthens the positive class impact on the difficult samples. So we also identified the influential substructures using impurity-based feature importance analysis, which is a feature importance score with a model that we have the best performance is our GBRT CFT4 model. So looking at the feature figures, we found the influential substructures such as the aromatic rings, the carbonyl group, organophosphate group. You can see in the corner here, the phosphate groups. And double bond CH and the nitrogen-containing heterocyclic groups here. Okay. So in conclusion, the developing various state-of-art machine learning models can help us achieve the best prediction performance, which are important in the computational toxicology. So all three papers I talked about were published and can be found on my own website. So the last part is we're also doing some work in response to the environmental health data ecosystem. We did a work in knowledge-driven discovery, which we constructed a cohort network, a knowledge graph that's towards the open science and knowledge-driven discovery for the cohort studies. We also developed a model to predict the coronary heart disease to aim for the precision environmental health. We developed an autoencoder survival analysis model to accurate prediction of the coronary heart disease in a population of American Indians. So this I will not be able to share today, but happy to share in the future. So going back to the open slide on the topic of harnessing high-dimensional exposure and omics data in the environmental health sciences. So there are much work we already did and can do in the future. I look forward to exploring more into the subject. So finally, I would like to thank my mentors and the collaborators, my postdoc mentor, Dr. Andrea Baccarelli, my PhD mentor, Dr. Wei Zhang, and my collaborators, Feng Gao, who have developed those we have developed these machine learning models together. Anna, Howie, Marianne, Yang, Tess, Allison, and the members of the Baccarelli lab, as well as our collaborators from Michigan State University, Dr. Hui Li, Lily Reister, James Tiji, and my colleagues. I would also like to thank Dr. Hannah Lau from Dartmouth for the collaboration in the microbiome studies, and Larissa Taxer, Virginie from the gestation environment cohort. I would also like to thank for the HPC resources, especially for the Michigan State University High Performance Computing Center, which is very high throughput, easy to use. We have also been doing some data transfer, large data transfer through the AWS S3 bucket and some cloud computations through the Ronin platform. And a couple of fundings to Dr. Baccarelli and EHS fundings. So for more information, you can find that on my website at ecosystem.github.io with the description and the link to individual papers, as well as the code for each paper that I led. And feel free to email me with any questions or follow me on Twitter. So before everybody go, I have one more slide. So our department, the Department of Environmental Health Sciences, have launched a new environmental health data science MS program. So this is a 12-month MS program for the training in the environmental health data science. So I created a group that you can scan this QR code to join. So after we join the group, we will have an October information webinar and a Q&A session with our chair, Dr. Baccarelli. And for more information, you can find them on our website at the bottom here. So that's it. Thank you very much. And I'm happy to take any questions.